All right, so I don't know if I'm in a, in the camera. So welcome everyone. Pleasure to introduce our speaker today. So Guillaume Blanchet from University de Sherbrooke. So I could say Guillaume is an ecologist and this is an ecology talk. But I think that's just a QLS talk because Guillaume is a prof in biology, in math, and in health science at Sherbrooke. So he's really like embodying the QLS spirit at Sherbrooke. So that's really great to have you. But the talk will be ecology. It will be. But so Guillaume got his undergraduate degrees and his master's in biology. So he's a biologist at the University of Montreal. But he got his PhD in conservation biology in Alberta. Who was your supervisor?
So
Okay.
yeah,
Okay.
in renewable resources, so it's not in the biology.
Okay, all right, yeah, okay. And then Guillaume went for a postdocs at McMaster in math, and there you work, I guess, with Jonathan Dushoff,
With Ben and Jonathan
Ben
and
Bolker.
David, yeah.
David Hearn, and the trio at McMaster. And then you went to Sherbrooke for a postdocs with mostly Dominique, I guess, Dominique Ravel,
Yeah.
who gave a talk at QLS already. You know Dominique. He's joined Sherbrooke in 2020 during a pandemic.
Yeah, it's been interesting.
Yeah, that's it. So you probably have stories to tell about this. So and Dominique is leading, if you go on this webpage, his lab is full, fortunately. So we have too many students, too many grants.
Yeah.
And yeah, sorry to hear that. But today's you have an ecology talk and you will tell us how to put data into meta-community models. That's really forward to this.
Thanks. So just to know a little bit, who is ecology? We'll go like that. Okay. So, that's not too bad. I guess there's people online too. So I figured, I was told that you wanted something that was quantitative, that was broad. So I took ecology as a topic. And one thing I think is interesting with those things is that I'll start with the basics and we'll kind of build up. So what is community ecology? Because the talk is about metacommunity. So what's community ecology? So if you're wondering, if you pick a book like this one here, and you look, that has a nice cover with a bunch of species on it, and you can think of a community as a group of species that occurs together in space and time. Right? That's kind of the general gist of what you do, and whatever is in that book cover. It's very nice, but it's not realistic. It's not true that you'll have giraffes and actually koalas and things like that. They don't live together, but it makes a nice picture. So since my talk is about metacommunity ecology, if you pick a book like this one, so that's what you would argue is the Bible of metacommunity ecology. At least it has everything in there. A metacommunity is a group of community linked together in space and time. So to give you an idea of what that means and how you can visualize this, I gave a few examples. Oh yeah, I forgot. So maybe before I give the example, what structures metacommunity? So the idea behind metacommunity is really, no worries. The idea is that there's essentially four big things that structures metacommunities. There's dispersal. So basically, individual will disperse in different ways. There is environment, so whatever your environmental setup, your habitat, the habitat of the species will be, it's going to have an impact on where a species can live or not. So if it's very dry, very hot, very cold, very wet. There's also the interaction between species. So they sometimes help each other, they eat each other. So accounting for this and dealing with that. And there's also evolution. So how the genetic makeups of species evolve through time, that can have an impact on the structure of the community. So for this talk here, I'll focus on those three aspects. I won't talk about the evolutionary part, but I think that will be enough to give us some nice things to think about. So a few examples. So I gave a few that I picked out of the literature. So just to give you an idea of how it works, you can think of, when I talked about a community, it's one group of species. And if you look carefully, you'll realize that there could be of different size. The idea behind metacommunity is that you really have patches where species live, where things live, and they're connected by dispersal. And sometimes they'll have the same species. For example, this mushroom there is in the two places, and sometimes not. And that's because there's different characteristics to those patches, and different species that live there can promote or hinder the whether a species will be there or not. This concept has been used in other fields, like in microbiology, has been proposed. If you think of different mice that have microbes in them, and you can think of the different microbes as organisms that you're following, at the species that you're following, and depending on how the mice interact, you'll have the dispersal event between the species. So each one of those mice is a community, and across this we have a meta-community. The idea behind the extra bit, if you want, when we're dealing with microbiomes, is that there's also things that kind of floats around that you need to account for somehow, some way. Another thing that has been proposed is to think of parasites meta-community. So different fish have different parasites, and depending on where they live, what they eat, and so on, and how they interact, they can distribute and it can kind of build a meta-community. Okay, so there's generally two ways to think of meta-communities. There's the theoretical way, and there's the empirical way. So, and those people that work in those two fields generally don't talk very much. And that's what my talk is about, is try to find a way to make those guys talk. So just to give you an idea of how people in the theoretical ecology field, I'll give you an example of a theoretical ecologist, which you know. And the general idea behind theoretical ecologists is that there's things that they know, and we know about the system, that is, and we can kind of narrow it down to this kind of quote, which is that, you can better understand nature through mathematical model that you define reasonably well, because you know the system and you look at the mechanisms that are there, and by looking at how those mechanisms behave, we can understand better what's going on. And now, if we take an example of an empirical ecologist in the field, with some colleagues in that case, which I guess you know, the idea is very different. Typically, empirical ecologists will go in the field, they'll try to understand nature by sampling it, by analyzing the data, by interpreting the results in the data, and making sense out of this. If you want to picture it a little bit more, that's how people see things. So theoretical ecologists will see the world like that in math form, and empirical ecologists will see the world a bit like this, where they'll try to figure out what's going on by sampling it, by studying it. And so very different perspective, and making the link is often done through statistics, and we have to be kind of careful and precise about how we're going to do this. And that's what I'll try to show you, at least one way to think about that. So I'll give you a bit more about the classic approach of how it works. And this is a nice picture to show you how to think about it. If you think of all of those lakes as different patches where there's species that live, and there's multiple species that live, well, those lakes at a certain time, they'll be disconnected. So like this. And at other times, when, for example, in the spring where the water will be slightly higher, the ice will melt, then they'll connect, species will disperse, and then they'll, when it gets warmer, the water table goes down, if you want, and then you'll get different different communities, and you know that they're linked somehow, some way, and sometimes more or less than others. So the way people have studied this, sorry, is through something called the variation partitioning approach. So the idea behind this, and that's from the original papers in Metacommunity, the idea that was proposed is that, and just for you to visualize it a little bit, I need to check time a little, yeah. To visualize it is that if you make an analysis and you just calculate how much variance is explained by spatial variables, by environmental variables, and the combination of both, we can kind of make get an understanding of what type of general mechanisms your metacommunity is structured. So the idea is that to visualize it, the big rectangle there is the variation across all of the metacommunities. So typically in that field, you'll think of a matrix where each row or each sample is a community where a bunch of species were sampled. And we'll have spatial variables, which I'll keep kind of vague for now, and some environmental variables. So spatial, typically, you can think of like XY coordinates and latitude, longitude, and things like that. But it can be a little bit more complex, like polynomials. And environment would be habitat characteristics. So what people have done is that they said, depending on which fraction of the variance is significant, because you can test them, then we can infer different types of metacommunity. So the idea behind this is if only the environment is significant, so if you only have this part here, we'll say that it's the species sorting mechanism that's driving your metacommunity. If it's only space that's significant, then we'll say it's either a neutral dynamic, like neutral theory, or patch dynamic. So these are ways to study, just to keep it broad. These are theories that have been proposed that assume that environment is not important, essentially. And if both are significant, then we can kind of guess that it's either species sorting or mass effect, which are, again, two different bits. And you can see some issues right away within this problem. Within this way of thinking is that species sorting appears 2 times because we're not really sure what's driving it. And the idea behind this is that there could be environment that's spatially structured that you didn't measure and things like that. So starting to think about this and the way we can work with this technically, if you're interested, what people have done is to look at partial canonical ordination and to test the different fractions. If you're very, very technical, you might wonder why you can't do anything with the middle one. Nobody figured out how to do it. It's actually a very technical thing. And it kind of drifted towards, we can't do it because of degrees of freedom problem, but I won't drag on this. So the idea and the main thing to keep from that part of the literature, which is about 20 some years old, is that basically the focus is at the metacommunity level. We can look at the metacommunity and say, you know, it's those types of mechanisms that are driving my system and kind of keep it there. And the question I was asking is, can we do better? All right. And so one way to look into this is to take a more refined perspective. The first thing that's interesting, if we keep that paradigm, is to include, in addition of environment, in space, to include co-distribution. So the idea, if you're not an ecologist, is that species are in a particular area because environment is important for them. It makes them survive there, because they disperse there. So that's the spatial part. And because there's other species that are good for them or bad for them in this area or around that area to make sure that they can live. So we figured, well, let's actually find a way to include co-distribution. And this is what, and I actually suggested that paper to you guys, and we figured out a way to do this and to go a little bit further. So that's what I'll talk a bit about here. It's to include species co-distribution and then to quantify the importance of each species and of each sample. So, all right, to include co-distribution, the way you work it out, and I'll kind of boil it down to a bit of the math here, since I figured you know a little bit about that. You're interested in this. Imagine that you have your site by species matrix. So each species is a column, and each sample, each site is, you can see it as a community where you sample your species, and you have a bunch of environmental and spatial variables that you sampled at those locations as well. So typically what people have done, what you can do, I'll say it like that, is that you can build a regression model, a linear regression for each one of the species. In sequence for each one of the environmental and spatial variables, depending on what you want to do on the distribution of species, what type of data you have, you can typically use a linear model or generalized linear model, like logistic regression or something like that. For the sake of the presentation, I'll keep with the linear model, but we can extend it if you want to talk about this. Feel free. I've done this a lot. And what you get at the end is a matrix of parameters, where basically you get weights on your environmental and spatial variables to tell you for each species what are their importance, essentially. Now, what's interesting about this is, if this is kind of all independent right now, but if we want to glue this together, we'll make the assumption that there's actually a common information attached to those things. So the idea is to say that mathematically that those parameters, they actually follow a multivariate normal distribution. And if you're not familiar with normal distribution, so normal distribution has a mean and a variance. A multivariate normal distribution has a vector of mean and a covariance matrix. And what's interesting is that those are parameters that are interesting to us typically to understand better what's going on, but those parameters too are relevant and they actually are meaningful. So this one here is the response of a typical species to the habitat into the space in that case. And it's a typical species is not something that necessarily exists. It could, but it's a conceptual thing. It's how my species react to environment in space. And this covariance matrix is, you can kind of see it as how a species or my typical species varies in response to one or a pair of habitat variable. And one would be on the diagonal because that's the variance, and the covariance would be a pair of habitat. And so that's actually a nice way to think about your system because it gives you parameters that make sense both at individual species level, but also at a community level. If you're interested in knowing about this, this is a paper that kind of introduced that perspective that's about 15 years old now, but it presents quite well this way of seeing it. Now, so that's nice. That allows us to kind of include space and environment. If we want to include co-distribution, then move things around a little bit, we have to account for something called latent variables. So the idea here is that there's stuff that are not measured by environment and space in my species that are either co-distribution, so interaction between species, or part of the co-distribution is environment that hasn't been modeled, or that's impossible to include as explanatory variable. So the idea behind this is that you include in your model variables that are good at capturing the signal that's still in the data that wasn't captured by environment and space. Latent variables are great for that. And you can generate them and you can weight them to give them importance depending on the species that you have. If you want to know the math behind this, it's in this paper. I'll be frank, this is not a light paper to read. Whatever is in Biometrica typically is not a light paper to read. But in that case, if you're interested in statistics, the idea that they propose is not only a way to generate those latent variables, but to decide on how many you should have. And they have a Gibbs samplers for this that they propose in there. Okay, we can talk more about this. I had fun in implementing it at some point in my life. It was, I'll say, joyful. I don't know if you're into that. So what's neat about this is that now we have within this framework a way to include environment, include space, and include co-distribution into our model. And while using latent variables. What's also neat, if you want to see the link with co-distribution, is that if you do a bit of math here, if you take some of the latent variables here, and you do a bit of matrix algebra, basically a matrix multiplication, then you can get a correlation matrix that allows you to get the information about how each species is correlated with each other species. Now, and that's the interesting part. So it gets us kind of an information about how those things are related. What's also interesting is that we can use that matrix there to, for example, present them in different ways. People have typically presented this matrix directly, but you can think of ordination plots where you basically take eigenvectors of this matrix and study them. So if we get back to this, looking at this particular framework, we can build up on this a little bit and start using the different bits to explain the variation into our model. So if I take that equation here and if I kind of redraw my co-distribution, my variation partitioning plot. So again, here in that part, we have environment and the spatial component. And in this part here, we have the co-distribution. Now, technically, what happens is that the latent variables are actually so good at, well, I'll say it differently. To calculate a variation partitioning analysis like this one, to get all the fractions and all the different bits, what you need to do is you need to build a model where you only have a spatial component and then one with only the environment and then only with the latent variables and all the combination to get all the different fractions. And This is problematic because latent variables are so good at capturing signal that they'll actually, when you leave them alone and you have no environment in space, they'll model everything. So because of this, when we got that paper that some of you read, we decided to split things a little bit and to say, let's just focus on, for the latent variable, on the things that are completely independent, the new stuff that's explained. And using this way of thinking, then we said for environment and space, to make sure that we get the right bits, we just split everything that's explained by both in half and looked at this. Small parenthesis. We're looking right now, we're writing a paper with a few colleagues that were involved in the original paper, namely Florian Hartig and Pedro Perez-Neto, to try to figure out if that was actually the right thing to do. At the time, we did it and said, you know, we think that's okay, but we didn't look at the details of the math of what that involved. So we're looking into that right now. Okay, so presenting this like that is interesting, but we figured it'd be nice to use triangular plot or ternary plot to understand better where things are positioned. And that's kind of a nice visual to present our results. So I'll present a few of those results of what they mean, of what they make sense out, and what we'll try to figure out where that can lead us. So that's one way to include co-distribution. If we want to quantify the importance of each species, because remember, when I looked at metacommunities originally, we only have information on the whole metacommunity. So having information on the species individually becomes interesting because it can tell us how each species is structured, what's the driving force. And if we have information on the contribution of each sample, then we can say whether a particular sample is more driven by environment, if it's more neutral, if it's more driven by space, or if it's really the combination of species that's important there. So to quantify the importance of each species, well, we have to use R-squares. And R-squares, this is like the classic definition of an R-square for each species. So J is a species and I is a sample in here. And the Y is, that's my data, that's my model. And this part here is is kind of an average over all the species. So over all the sample, sorry. So we can calculate that. And that's very straightforward to do in the sense that it's easy to calculate because we have our model and we can just plug in that equation. If models are not built from linear models, they're more built from generalized linear model. There's specific R-squares attached for this. This is something I don't want to drift into, but we can again have a talk on this after. The short answer is that we can basically build, locate, so do the variation partitioning by species and see where they are located into our ternary plot. And we can draw them like this. And what's also interesting is we can get a community level, if you want, or a meta-community level, R-square, by just averaging across the species, and we get a position of what the meta-community is. So things are, the idea that we had previously does kind of translate. So the original meta-community theory translates into what's going on here. So that's very nice. If we want to look at sample, then we have to be a little creative. So if you look at the R-square here, we realized, and actually I'm not going to get involved in this. The story is interesting. I was with Matthew Labeld and Pedro Perez-Neto, and Matthew Labeld is a professor at the University of Florida, and he's a guy that has great ideas. And he said, but he doesn't know how to implement them. So he said, you know, it'd be great if we can figure out how to contribute, how to look at the contribution of each sample. We said, yeah, that's true. And how do you do this? So I have no clue. You figure it out. You're the quantitative guy. And Pedro, who's very, very comfortable with, Pedro Perez-Neto, who's very comfortable with R-squares, said, you know, I think I have a way. And we kind of scribbled down things where it's just a way to decompose the R-square in different samples. And a bit of one standardization here with the 1/n to make sure everything's all right. And then we can get, because we remove the sums here, we can get a unique, if you want, sample level R-square. And actually for each species. And if we average across the species here, because we're interested in the contribution of each sample, then we get kind of the contribution of each sample to the meta-community. And we get something that looks like this. So these are all caricature. And what's important here is that if you look at the values here, because they're decomposition, then the contribution, you basically, when you sum them, you get the R square for the species, then they're all typically very, very small values. And that's normal because you just split them into across the samples. So to show you a bit more of what that can entail practically, I'll give you an example that we're finishing up with a study on passerine birds in Montreal. The idea here is that we were interested in studying the major green areas of Montreal and seeing what are their importance in structuring the bird community, the passerine community. So essentially, this is our meta-community where we have all of those blue patches here, which are the green areas. There's 44 in total that we considered, and see how important they are for the passerine birds. So to give you an idea what we have, there are areas that are very large, that have kind of agricultural nature. So if you're never been to the western part of Montreal, there is agriculture there. There are parks that are, I'll call that, well, they're not parks, they're green areas that are kind of left to themselves up to a certain limit. And there's parks that that are not far from here that are more, some examples that are more used for leisure. So there's kind of all of that gradient that's not necessarily spatially structured. And for the species, what we did is that we focused on 1 year on the nesting season for 106 passerine birds using, in that case, census data, where we have all the species across those parks. And we focused on this area. In that particular presentation here, I decided to focus only on this period, but we're actually the final studies comparing both the nesting and the non-nesting period, because we were interested to know if the distribution was different across those different periods. Short answer is there's a difference, which is actually quite neat. So for what we used for environmental variables, we used the size or the area of green areas and the perimeters. For spatial variables, we use distance-based Moran's eigenvector maps. So, okay, so if you're not familiar with this, these are essentially spatial, it's a spectral decomposition of XY coordinates. I don't want to go too much into the details, but basically they allow us to get waveforms across the space to get the spatial structure. They're typically, when you build Moran's eigenvector maps, sorry, you get as many variables as you get as you have samples minus 1. So you need to filter them. And in here, I'm trying to remember, we kind of selected them based on how important they were into the model. So there was a variable selection procedure. And what's interesting there is that they're all independent of one another. So in the sense that they're mathematically independent or orthonormal. So whatever is explained by one it's going to be completely independent of what's explained by another. So we get a spatial structure. And we have the co-distribution that's included in there. And that's the type of result we get. So to walk you through this a little bit, so that's the importance of each species. So each of the bubble here is kind of related to the size of the R square. And well, what that means in practice, that means actually a few things. The first thing is that there are those species here that are different from the others. There's a few. They're all rare species for Montreal. So if you're a birder, then you might know that some of those species are not typically well adapted to Montreal, or they're not typically found here. So this was interesting in itself. They're essentially rare, so it's hard to figure out what's going on. And they are also species that are not well adapted typically for city environment. At least that's what we figured out. The other thing that's interesting is that, and that's something that kind of flashes very quickly, is that environment is a huge driver for this data. So that's kind of interesting. not totally surprising, to be frank. And the other thing the last thing that we thought was interesting is that all species are kind of important. There's no species that have a very small value, very small R square. Even like they look like they're piled up, but if you look at the results in detail, you realize that they're all kind of important. So these are the type of results you can get. So you can go dig deep in not only understanding the general structure and relating it to some theory that's a little vague when you look at the metacommunity structure, but you can also look at, get a bit more of an idea of what the drivers are. Okay. And if we look at so that's the species side. If we look at the sample size, then we get something like this. So what's interesting here is that I don't know if the screen is great, but you get very, very small points that are kind of scattered everywhere, and you get one in the corner here that's huge. And this one is actually this big area. And again, this is not super surprising, and this is the reason why we're still working on it. We want to know what's going on here a little bit. But in short, that particular area contributes a lot. And again, this is not something that should be overly surprising because it's so big. And we know that this is ecologically, it's known that it's important to have big areas. In that case, it was kind of a surprise at how much important it was for the whole island. And the question we were asking then is, all right, this is all neat, this is fun, but can we do better than this? Can we go deeper? And so now I kind of walk you through all the stuff that's been published, and we're kind of drifting into the world of we're getting there, and we're trying to publish the last bit. So to understand how we can better link what we've seen so far with what I'll look into right now is take a step back and try to figure out what are the mechanisms or what are the models that we can use to understand how species compete locally. And if you're familiar a little bit with that literature, and if you've even if you're not very familiar, typically you've heard about this type of model, the Lotka-Volterra equation, which is, it's a model that's been studied a lot, that's been presented in many ways. Maybe that's not the formulation you've seen. So if you've seen it, so the idea is that the abundance of one species here at a particular location will vary regarding in relation to a certain intrinsic growth rate and the interaction it has with another species, essentially. And what happens typically is that you'll get something that looks like this. It's either going to stabilize at some point, or it's going to continue, the wave will continue kind of overlapping. In short, we can do that typically it's done for a pair of species when you learn it in the base, your first classes of mathematical biology, but you can kind of build that with as many species as you want, with alpha becoming a big matrix. And people have been very crazy at kind of picking at it from different angles. But the idea here is that if we understand well what's going on, the mechanisms of how things are, we can implement it into this model and study how it will affect the local dynamics of species. Now, if you think regionally, so at a slightly bigger scale, then typically what people will do is they'll look at the metapopulation model. So, so that's the work of mainly Richard Levines, and Ilka Hensky developed it quite a bit. I did a postdoc in his group at some point, and it was very interesting to see how people think in that group. Typically, you don't see this equation like this, but this one will become it's interesting for us. So typically what you see when you study metapopulations, you're saying that what's colonizing a patch and what's making a patch go extinct, so the species in there will either colonize a patch or the patch will die off. But you can also present it as isolation, so saying how far away a patch is from the other one. And that's a measure up to a certain limit of colonization. So just to walk you through this, here we don't have the number of individual, we have the probability of a patch being, or in that case, of a patch being occupied. And this depends on extinction and on isolation, and isolation is a component of colonization. Now, if we understand this well, And we know that what happens regionally depends on what happens locally. So in the sense that if you're looking at a patch, like you're all here in a room, then you know that your neighbor will have an impact on you. And whatever's happening in the other room will, you're gonna go there at some point. But it's all the interaction that happens between individuals that are important locally and that may impact what happens regionally. So we said, it'd be fun to basically try to figure out how to put this into this. And so we figured that one way to do this, because whatever happens regionally depends on what happens locally, means that those parameters here, so isolation and extinction, they are things that are actually, they can actually be functions of what happens locally. So instead of having single values, single parameters, we can say they're a function that depends on abundance. Okay, so these are function. If we start by building a model that looks at equilibrium, then solving this at equilibrium gives you this equation, which is a very, very annoying equation to work with. So because if you want, and the idea again is to link with real data. If you want to link with real data, you having something that looks linear is a good start. So after a bit of pondering, a bit of thinking and looking at the literature, we figured out that applying the logit transformation on this actually gives us something like that, which is much closer to a linear function. And that's something, at least for me, when I looked at this, I said, I can work with that. And the question now becomes, what fits into those two things? What's isolation? How can we find isolation and extinction from specific data, from species? So if we start with extinction, extinction is a little bit more easy. From a, just gonna look at time, yeah. What's interesting about extinction is that we know reasonably well what are the main drivers. So typically, what are the drivers of extinction is that basically environment is bad, or there is new species that compete with you that makes it more difficult for you to live in that particular area. So statistically, what that means is we can actually write this down using some of the same the same notation as before. And we can say that, the X will be the environment, beta becomes the parameters that weight out this environment, the importance of this environment. And we can have a measure of the importance of interaction or co-occurrence, but I kind of use interaction here because we're linking it with, from a theoretical perspective, although we don't have measures of interaction. And because typically, because we're working with local information, then we're counting stuff. We're counting how many species there are. So statistically, that basically amounts to, if we want to find the expectation of extinction, then we're assuming that we're following a Poisson distribution or a negative binomial distribution, which kind of force us to go with using the exponential link to get the expected value. So we can get to that, and that's, from a statistical point, reasonably easy to do. Now, if we look at isolation, then we had to think a little bit more. And the key came from this paper here by Ate Moilanen and Neil Kahensky, where they define isolation as something that depends on the migration. So they call it a migration rate. So each species will be, and now just to make sure, I just realized migration in the metacommunity perspective is essentially how good you are at moving between different patches. It's not migration in the sense of species are migrating in different, like birds are migrating for the winter season. It's really a different wording, and this wording is how often, or how comfortable you are in traveling between different patches and going between different rooms, if you want. So, and the idea is that each species has a different migration rate, and that will depend on the distance between the different patches. So if you're far away, if the patch is very far, then it tends to be more difficult to reach. And the size of the patches, because if it's a very big patch, then it's easier to reach. And they included in their model a beta to say the importance of that size for that particular species. And all of this depends on whether a patch is occupied or not. So if a patch is not occupied, then it can't kind of disperse different species. So our task when we did this was to figure out a way to take this very nice theoretical idea and to try to kind of dig it into the stats. So we came out with something that looks like this, where the idea here is using the latent variables that I talked about a little earlier to say that for a particular species, if a patch is occupied or not, we'll give it an importance. So that's a matrix here that has diagonal elements where if a patch is occupied, it's a 1, if it's a 0, if it's empty, it's a 0, and everything else is 0. By doing this, if you've played with matrix algebra a little bit, you realize that you basically just get an empty value if you have a 0 on the diagonal, and then you just get the same value if you get a 1. And that turns out to be a nice way to look into it, to just say, is basically my distance, my patch important or not? And on top of that, if it's important, then it depends on distance. And in that case, the migration rate. So we'll basically say that this parameter also depends on this, has a spatial component attached to it. So again, we assume, because we're counting stuff, we're assuming that it follows a Poisson or a negative binomial distribution. And that's where the exponential here comes in. So kind of walk you through the hard part. Now, if we take the two things that I've discussed to define the function that defines isolation with data, that define extinction with data, and we plug it into our model, we get something that looks like this, which is rather ugly. But the cool thing is that it simplifies, because logs and exponent, they cancel out, and we get something that looks like this. And in practice, this is fun, because we have a model that basically the parameters make sense and that we can estimate, because we have all the tools available for that. So just for you to understand what I mean by the parameter makes sense, well, this parameter here, so the lambda tells you the importance of space in defining isolation. This matrix M here is just, it's basically based on data, so it's not a parameter. This parameter here is the importance of environment in driving local extinction. This is the importance of interaction. And this thing here, through this, we can estimate a migration rate. And now if you're very, very keen, if you have very good eye, you realize that there's basically a sign shift here. That is something that is very okay. It's okay to do that because it's basically the interpretation we make of those parameters here. So we can just shift them and it's okay. All right, to give you an idea of how that works in practice, so I used an example where we sampled, so there is data that, since you mentioned Dominique at the beginning, so with Dominique there, he has been working on every year sampling a part of Mont Satin, where he has a kilometer by 200 meter transect, where he knows, well, we know all the trees and plants that live there at a reasonably precise location. So I think we have like something like 30 centimeters air. So if I split this into quadrats of 20 meters by 20 meters, actually within each quadrant they have, within the whole area they have, they sampled all the trees, but within each quadrant, within the center of each quadrant, they sampled, I think, within a range, I think it's a radius of 3 meters, all the plants. So I use this as my meta-community. And so there's 72 plants. We know that environment is, like elevation is very, very important. So in that example, I use as environment elevation squared. So the idea of taking elevation squared is that you basically, if you center elevation, then you get kind of a humpback variable where, or a quadratic variable where basically you can capture whatever is in the middle and not in the edges. And we kind of passed it through the model with focusing on that particular species. Veratrum viridae is a species that is known to live in that area. So just to show you the visual, each quadrant where it was found is in black and all the other ones are white. So if we build that model in here, the type of result we get is a map that looks like that. So that's the probability of occurrence of my species. So it's within R square of 0.417, which is neat. Actually, it's the best model we got. We were quite happy. I found that fun, but oops. All right, sorry. It's okay. The most interesting thing about this particular model is not that it's good at fitting the species, but that actually the parameters make sense. So, like I said, we can, in that case, say that elevation is an important driving force, and it's, sorry, and that basically there is kind of a gradient that goes from top to bottom, but mostly there is something in the middle. So that species is not found at the bottom. It's not very much found at the top. It's mostly found in the middle. You can see every so often, but it's mostly found in the middle. And we can see this by this parameter here. And again, that's the sign that you see here. The negative sign is where this comes from that I mentioned earlier. We can have measures of interaction. So some species were found to show positive co-occurrence, and some show negative. And if you're fast, thank you. If you're fast at doing math, then you'll realize that 49 plus 17 is not 72, because there's some species that basically show no relationship. We found that there is some measure of isolation of migration, but mostly not very much, because the parameter that drives it in front is not really important. So basically migration is not important. Dispersal is not a driving force in this case. So it gives you an idea of how far we can go to understand what's going on with a model that's like that. So in conclusion, and that's all with hierarchical models. So it's possible to link theory with data and to actually make each parameter make sense ecologically and link it with theory. They're interpretable, so that's cool. However, and I want to stress this, I'll be careful. Okay. We need to be careful about the way we make sense of those parameters. So I use interactions, and I tend to be very, very careful about this. We don't really know, because there's things that we didn't include into our model, but since we can make the link with theory, we can kind of make inference and go deeper at some point with this. The next step is that if you talk with empirical ecologists, they like to sample and they like to be very creative at gathering finding different ways to gather data. And so whether if it's microbiome or nanosequencing or eDNA, there is kind of a huge effort to change the way we gather data. And adapting our models to this is important into other systems as well. So with this, thanks. And these are the two people that were involved in this project.
Two questions. The first one, I may get lost. What a latent variable you mentioned?
Yeah. So a latent variable is a variable that you didn't measure, but yet that you constructed to capture information that wasn't captured by the other one.
So it could be any other variables.
It's any variable that is not sampled. So if you want like an example, you know what principal component analysis is?
Yeah.
So that's one way to think. Like one axis of a principal component is a latent variable, because it's a combination of whatever is in the data that has signal that you want to capture.
Okay. The second question is the example you've given for the birds on the Montreal Island. Because I also study in meta ecosystem. I was very struggle in implementing the experiment and theories. So for that example, I think when I build, if I were building a model, I will struggle at the birds the bird do not only live on the green areas, they also live in the cities. So
yeah,
they will there will be an output from the meta communities.
Yeah, actually, that's a good point. That's why we use the nesting season in here, Because in the nesting season there, well, the original idea, so I'll expand a little bit. So you're very right. Birds tend to move. So, and what happens is that in the next nesting season, our hypothesis was that they actually will compete very much within that area. So they want to fight off their area and protect it. So that was the idea. And what we did is in the full studies, we said, if we know what happens during that period and a little bit outside further in the summer, then we can get a better idea of if that's true or if they are always kind of protective of their area. And it turns out that they're actually a very big difference during the nesting and non-nesting season. So you're right, but they do kind of have a reasonably small dispersal, except for some species which weren't included. That's why I focused on passerines. Like if you take pigeons and crows and things, they just go all around, and they're kind of irrelevant there.
The following question. Do you think the input or output is important in building the meta community?
But what do you mean by input and output?
So we build the meta ecosystem, there will be input and output for like matter or
Okay, yeah, I think I know what you mean. It's a different angle you take on it. I think if you take that perspective, they are important, but it's hardly, my experience, maybe I'm wrong there, maybe I haven't seen someone taking really that perspective specifically, but maybe I'm wrong.
Maybe you're talking about just immigration from
between patches, like out of the patches you consider.
Yeah, that's it from outside the system.
Yeah, like from lava to Montreal.
Yeah,
Okay.
That's what.
Okay, now I see. So short answer is yes, people have done that. And in here, I haven't looked into that in that particular example, the passerine example. But yes, and the way that I've seen people do is just basically there's kind of there's a big area that doesn't exist. That's a conceptual thing that just things move in or move out, but not any more than that. So it's kind of a migration parameter essentially.
So, thank you, really cool talk, appreciate that. I'm curious I would love to hear you go a little bit more about like your very last point on the limitations of the interpretation. Also connecting to, we read two of your papers and one of them was mentioning how due to all of these different factors, it's really hard for us to infer like species interaction from just going on this data, right?
Yep.
So, we were discussing quite a lot of that, it was really cool, like.
Can we save that question can we go with the first question and then save that for when we're in a group discussion?
Sorry, yes, I was just thinking like, because that may be, I don't know, just for me, why I want to ask that question, yeah.
What, I'm confused.
I'm sorry, I'm curious about like the limitations and how you, like what informs you on what limitations are to interpret these models.
Okay.
So, for example, what you mentioned in your first paper.
Okay.
Yeah.
So the limitations, this is kind of a broad question.
Yeah.
And I'll try to keep it, considering the room, I'll try to keep it broad. I would say, I'll say it like that. When you work with data, remember that this stuff was sampled and it's not perfect. And that creates, and often when you play a lot in front of a computer and you kind of forget that there's error in the sampling, there's error in your model, there's things that are not perfect. Thinking of that is important. So I have a very quantitative lab. I tell all of my students, even the statisticians that don't want to go in the field, to go out to do experiment a little bit to understand what's going on. So that's one limitation. And these are because there's basically limits to your data. So that's one thing. The other thing is that there are limits to the interpretation you can make of your models because your models they're all wrong, right? I mean, they're not. There's always problems. They never get to the details to fine-tune details. So you want to be careful about that as well. And in that case, I know for a fact that I should never interpret all of the things that I call interaction in quotation mark because they're not really interaction. they're basically stuff that wasn't explained by environment in space. But if you link them with the theory, you can make a link and say, my theory says that this should say its interaction. Is it? I mean, it's hard to know. There's something out there there is interaction, it's just hard to measure. So the way to get to that in practice is to basically go down and do other experiments to actually check if there are things that are interacting. So you see, there's kind of those different ways to look at your data and to look at your results to make sure that, you know, there's limits to the ecology that you can get to this, to the biology you can get out of your system. At least that's my take. Yep.
So for the model, I think this might be just, I don't understand. It's like, why did you decide, like, or how did you decide to split the factors affecting the distribution
So, okay.
that way?
So, all right. You're just to
I'm
make sure.
talking about like the circles.
Okay. So you're talking about, I'm just going to go back a little bit to make sure we're on the same page. This.
That one. How,
So why this way and not other ways?
yeah, and how also.
Okay,
in the
so I'll tell you the practical bit. The practical bit was this part, it had to be like this. Because so the way you calculate a variation partitioning analysis is that you take this circle here, so whatever, so let's say only spatial variables, you calculate whatever is explained by this,
Okay.
and then you kind of keep the R square there, and then you calculate only the environment, and then you keep the R square and you calculate both together, and then you do basically arithmetics to say, you know, what's to get this fraction in. Okay. And if you can do that for each combination. The problem is a technical one. It's that when you get to latent variables they're great because they get you to another part of your data, but they're annoying because if you have them all by themselves, they'll explain everything else. So because they're good at capturing signal. So when we saw this, we tried a number of things and we realized that the best way to move forward was to say, well, we'll just look at the whole of our model and controlling for environment and space, and looking at what is explained by that, by only latent variables. So that's why we focused only on this thing. Technically, we could figure out what those two are without the latent variables, but since we didn't know what portion it was, we felt it was safer to basically just split in two and go on. Now, that's the technical answer. As to why we decided to go there and not another way. Basically, when I was writing the paper, at some point I said, right, we need to fix this, and we're at the stage where we just need to get this out. And I said, let's just do this and be done with this. And let's see what the reviewers say. And reviewers were actually quite nice, and they said, oh, it's an interesting story. We'll see where that leads us. And the next step that we're doing right now is to basically try to see what are the different options, and it actually becomes quite technical in figuring out what is a measure of variance and how, because there's different ways you can think of variance. So that's the long story. That's what it is.
I have like kind of a follow-up question, I guess, because it's based on this schema for the Pasteurine.
Yeah, this thing here.
ternary diagrams.
Yeah, I'll get there. This one or the other one?
Well, first the other one and then this one. How do you interpret this in terms of co-distribution?
So well, there's not much, I'll say it like that, in the sense that like co-distribution goes like this.
Yeah.
So essentially what that means is that co-distribution is not a big driver.
Okay.
So if you had something that would be, just to give you, it would be all here, it would be that space is the main driver. And if it's all here, it's basically that environment is not important, space is not important. And so basically what you can say is that those 4 or 5 species, I think there's 6 there. There is some aspect of co-distribution. So that tells us that, in short, some of those species tend to be found together. Kind of every so often, a little bit more than the other ones. That's what it means.
And for the next one then,
Yeah.
so you have the big area is super important in terms of environment, but if it's because it's a huge continuous space, wouldn't it be a spatial variable? Did you?
It's actually no, because of the environmental variables we picked.
Okay, yeah.
So because it's basically that it's big and it's convoluted, right? It's not, it doesn't have like a, the idea to take the perimeter is that there's some of those patches, like, I don't know if you can see them well, but some are reasonably square and some are quite long, like this one here. This one, it's only about that. The spatial is about the area and the perimeter. If it was spatial, then basically what it would mean is that we would have like, for example, a lot of species here, very few species here, and potentially a lot of
Okay, so
species
it's
here.
spatial in terms of
The whole island.
limited okay, makes sense. Thank you.
I think you were.
Yeah, I have a couple, I guess like I'll limit, I guess, the questions, but I'm curious about these like ternary plots. They're super cool. I guess maybe I interpreted the one before different broad, I guess, but the species in the middle, so which ones are the rare species in these two clusters, the middle, right?
Yeah.
Okay, so I guess like how do we interpret like common species, let's say, a more abundant species being kind of towards like one of the vertices, let's say in this case environment, and then rare ones being in the middle? Like, is there a scenario in which like I guess the abundant species would be in the middle versus the rare ones on the what would that mean, I guess, in terms of like that specific environment?
Well, basically, if you had, okay.
Yeah.
Just to make sure I got that
Yeah,
right. Let's say it was the invert, like all the commons one were here and the rare
Yeah,
ones
yeah.
were there. What that means is that basically, what are the main drivers of that system is that the reason why you find species there is because there's other species there.
Yeah.
And or
Mhm.
that there's some environment we didn't account for that's very important.
Okay,
Like, and that could change, like if you think of, I don't know, hotspots, like hot zones or things like that, like that could be one of those things which we didn't include. It could also mean that the reason why those rare species are found in that hypothetical case with the environment is that there's specific environmental conditions that are unique. So, for example, the size of an area is very, very important for a set of species or one or two species, and we can only find them there. And that's the logic.
Okay, cool. I'm curious about, I guess, like a second question for like the plot with like the different samples.
Yeah.
Like, it's like, what's
This one here?
Yes, yeah. So I'm curious, just like either if you take like some measurements of the area or the perimeter for each of those patches, like if you kind of scale the R squared by just like would you kind of see, would it kind of be like like a nice fit, or would it be like, I don't know, like maybe like a keystone patch where you just explains a lot more variance than would be expected for that area? Like, is there any, like, I guess, interesting patch for they're kind of like outperforming on the R squared, I guess, than they would be given their size? Like, is there any like weird,
Yeah, I think there could be some environmental structure that's important. There could be, like I answered, like, if, for example, you have different sets of species across the island,
Mhm.
then space would probably, knowing that the environment is reasonably uniform, then it could be that spatial would be very important there. That's the way you would highlight that.
Okay.
And in that case, you would have something that would be more there.
Thank you.
Okay, I'm not an ecologist,
Excellent.
but I mean, the purpose, as I understand, the purpose of this project was to, test how good a model, a theoretical model can explain the probability of existence of some species in a patch. So, I wonder if, the probability of this probability is obtained by, some tools in chaos theory to get the ergodicity of this,
Okay, I think I know what you mean, in the sense that, is there other ways to get around that? Is that what you mean?
no, I wonder how the probability of these, obtained in the theoretical, how we obtain the probability from the equations.
When you only play with theory, you basically, you can just start with, so have you, I'll ask like that. Have you played with differential equations, like system of differential equations?
A little, yeah.
So Basically, it could be that your different initial values, it could depend on your initial values and see where that leads you. So you can decide on this, and if you, and that paper, the paper where we present, so I'll go back a little bit. But this paper, I don't know if you can see it well, this paper, I'll get it. No, anyways, you know what, I'll go to the end. This paper is, let's get it later. This paper there shows how to do that, because actually this is a theoretical paper. So where we present the theory or a theory of metacommunity, and we present the stat around it. So what we've done in that paper is that we propose different types of mechanisms that we know are there because we implemented in there, the model. And then we looked at how our stat was good to capture it. And so, and how our statistical models were good at capturing it. And we figured out that it actually works out well. How chaotic it is, actually, it's not very chaotic, because the underlying model, the metapopulation, the metacommunity model that we used in that paper is essentially, my co-authors wouldn't like me, but essentially a glorified logistic equation. All right, that has a little bit more to it than that, but it kind of always has, it always levels off. We can make it more complex, but that was good enough for us to get around. Yeah.
In the bird. Can we just, let me stop the recording, and then we're gonna have some pizza, and then we're gonna continue this whole discussion. So thank you very much, Guillaume. Thank you.